So first of all, thank you so much for inviting me, and it's my pleasure to be here and talk to you about molecular detection and the work we do in the lab. But before we even move further, I have to say everything that I'm going to be presenting today is because of dedication and hard work of a number of people. They are in my group, collaborators across the country, across different universities, but mainly these two individuals are my master's students. ML worked on molecular detection that was published in 2018, and the work I'm going to be presenting here is Tammy's master's thesis as well. So I will be basically focusing on Thousand Cankers Disease. For those of you that are not familiar, I'm going to provide a short overview of the disease, symptoms, how to identify differences between East and West, and basically the challenges that we have with morphological confirmation. So Thousand Cankers Disease is a complex disease that involves the host. All juglone species are susceptible, or all walnut species are susceptible to disease pressures. The fungal pathogen is Geosmithia morbida, which is the first pathogen in the basically genus that became pathogenic. This is the pathogen or organism actually that lived in symbiosis with other bark and ambrosia beetle, and it was not considered significant for the forestry in general. And the primary insect vector is Periopterus juglandis, or walnut twig beetle. However, in East Tennessee, we found additional vectors, and in some other states, they were able to find alternative vectors to this particular disease, making the confirmation and identification a little bit more challenging. The reason we care about this is simply because walnuts are economically, socially, environmentally important, and the current standing value of just eastern black walnut, which is the native species for the eastern United States, is over $569 billion. So disease is quite interesting. So you do not know that necessarily disease occur until the bark is peeled, and the cankers are basically being formed inside of the phloem area. So these cankers can originally be small, oval, dark brown, but they can coalesce, create these large necrotic areas, and eventually girdle the tree. The conidia, or the spores, are what I usually try to explain. It's almost like a pill-shaped like form. They can oftentimes create these chains, but part of the problem with that is it's a lookalike with a number of other species, making the morphological confirmation a little bit more challenging. The beetle, walnut twig beetle, is tiny. It's less than 2 millimeter long, and it creates these basically tunnels inside of the phloem area, carrying the Geosmithia more likely on the external parts throughout the tree, and that's how the pathogen spreads. So Thousand Cankers Disease has appeared in the late 1990s in the western United States, but it's spread throughout the country, and right now it's in Europe and Italy, but it's also a global concern because all juglone species are susceptible, and although we have a number of susceptible species and highly susceptible in the United States, none of them are resistant. And walnuts are distributed globally, so this could potentially be a problem beyond our borders. So current Thousand Cankers Disease distribution is basically across more likely entire western United States. The shaded area here is the native distribution of black walnut, and these are the TCD positive states, or Geosmithia morbida, the pathogen positive states. And the reason we have them divided like that is this is there is no necessarily federal quarantine or federal regulation regarding the pathogen. So it is up to states to decide whether or not you have to have both pathogen and the beetle to call the TCD positive county or state, or you have to have one or the other. For the state of Tennessee, we have to have presence of both, and that is the current situation just for the Southeast. Part of the issue that we're dealing with this is the differences between Thousand Cankers Disease severity in introduced, which is the western United States range, versus the eastern part. So in the western part of the country, the disease basically is manifested with die-off of upper canopy, yellowing of the leaves, and basically exit and entrance holes of the walnut twig beetle. And within 3 to 5 years, basically the tree dies. And this is in Utah. This is on a Colorado State University campus. You can see the arrows pointing here, upper canopy die-off. Unfortunately, although Arizona walnut is least susceptible under the greenhouse inoculation trials, this is one of those Arizona walnuts that is almost entirely dead. So although it's least susceptible to disease pressures, still the disease itself can be devastating to a number of trees. On the other hand, in the eastern part of the country, we had two completely different outcomes that we rarely see in the western part of the country, because in some cases we've seen, for instance, in Boulder, Colorado, almost 60% mortality rate of black walnut, which is, again, planted, not native in that region. So arrow pointing here is basically a die-off of these upper branches and yellowing of the leaves. But this is the same tree 3 years later. And it's very important to notice this is in East Tennessee, and in 2006 and 7, we had a really severe drought. And then all of a sudden, this is 2010 when the disease was discovered in Knoxville, Tennessee. And this is the same tree that was infected 3 years earlier, almost completely recovering. On the other hand, trees in the same area is not necessarily having any recovery, and it's completely dead, and the tree had to be cut. So we had these two very, very different outcomes. Trees either recover to some extent, and we've done a number of really interesting research, and I'm not going to go into that. What I'm going to, basically we found a number of trichoderma species that is associated, which is basically used as a biological control. We found that when we cultured number of isolates from the east versus west, California samples, that we found trichoderma samples in the eastern United States in larger quantities, which could explain some kind of genotype and host defense mechanisms, which is maybe one of the reasons that we're seeing differences in the eastern part of the country. And my lab took that idea to the next level, and we're doing a number of inoculation trials, trying to do biological control and some chemical control that was recently released. But in the meantime, what we wanted to do is basically figure out ways how we can do quick molecular detection, because morphological detection can take weeks. So the first step was to develop, this was in 2011 and 12, we developed microsatellite loci, or microsatellite markers, which are basically species-specific primers that were developed for the pathogen of TCD, Geosmicia morbida, and the beetle vector, walnut twig beetle, individually. And we're trying to see, can we develop certain methodologies and can we reduce the cost of this molecular confirmation. So the first thing we did is basically these are testing 4 different primers individually, but also because price per reaction is quite expensive, and if you want better confirmation, using more primers is always a little bit better, because sometimes not every sample is going to amplify in every reaction. So we're trying to figure out, basically, can we multiplex and combine more than one primer in a single reaction in a meaningful way where we can visualize that using the kayak cell. And yes, long story short is we can do that. We can combine 4 different primers in a single reaction and confirm presence or absence of the pathogen in this particular case. So what are the issues and challenges between classical diagnostics and rapid molecular detection? So the full credit here goes to Romina Gazis, who was a postdoc in my lab at the time. She's running disease clinic in Florida currently, and my master's student, Amel Oren. So when we received samples, the difference even from the wood sample from east and west was pretty striking. So the first step is look under the scope, try to find this, basically peel the bark, find exposing lesions, or some ways in which we can see galleries or mycelia forming structures by the pathogen. Then we take basically bark chips, put it in this basically petri dish, creating a moist chamber environment, waiting for sporulation. And then within 3 to 5 days, we would basically see something like this. So this is a conidiophore bearing conidia. This is the fungal structure, and ideally you would pluck one of these, plate it, and we actually did something by recommendation from Nat Tisserand, who was the forest pathologist working on this system out west at Colorado State University at the time, because this is very slow growing and quickly outcompeted pathogen. He did something very similar to bacterial streaking, which is not commonly done with fungi, but it worked pretty well for us because you can get rid of certain contaminants if you do this. So we do a version of bacterial streaking, but this is a fungal streaking on a plate to eventually obtain the pure culture. So the whole process can take sometimes 4 to 6 and even longer in the eastern United States. From the western part, we would get these samples much quicker and better. Unfortunately, the reality is that this is what we often see. So the cultures are quite often contaminated. You have other faster growing pathogens. Just is super slow. It takes several weeks to create a small colony on a plate. So we needed to find a better way of identifying these. Another part of the problem is that we basically have extremely big morphological plasticity. This is all the same pathogen confirmed using both morphological and sequence data, but it looks quite different in culture. So if you're not trained and if you haven't worked with these cultures for a long time, it can be confusing. Also, depending on media, this is an PDA, this is an MEA, this is something that looks very, very different. And under the scope, you can have lookalike cultures, lookalike conidiophores bearing conidia, which can be confusing, and in some cases you can misidentify the pathogen of interest. So to mitigate that, we wanted to develop a rapid molecular detection protocol. So this was the first step in that process. So we wanted to do twofold here. We are always thinking simplicity, reliability, and reduction of cost. So we wanted to do two different things. So one of the things is to peel the bark and expose the cankers and basically drill through this part of the wood and obtain what we call drill shaving, which is basically sawdust that we will eventually use to isolate DNA. The second part is to try to figure out the option of maybe using boots on the ground in the field, people that do not have to peel anything, but use natural opening lenticels, or if there is an entrance and exit holes from the beetles, they could basically just blindly drill in and maybe capture. If you do this multiple times per branch, hopefully we'll capture enough if there is any contamination, and use that to isolate DNA and see, can we do that without any peeling, without any visual confirmation. So that was the idea behind it. And also to capture the differences between east and west. The high incidence of walnut twig beetle is very prominent in the western part of the country. We talk about, for instance, in Walla Walla, Washington, where we work with collaborators from Purdue, we would have thousands of beetles versus several hundred in the east part of the United States. Very fewer galleries in the east and number of galleries in the west, and much harder confirmation of the pathogen in the east versus west. So we needed better identification protocols. So what we did, we developed those microsatellite loci that are specific to the species of interest. In this particular case, Jusmicea morbida and walnut twig beetle. They would amplify if we have a positive sample at certain base pairs. It should be in a range around 200 to 250. This way we can use morphological as well as molecular confirmation of the presence of the pathogen or the beetle. So what we did is basically we collected 40 trees from each location: California, which is heavy infested, Missouri, which is disease-free area, and Tennessee. And from each tree we collected around 10 branches. However, Tennessee, the trees were in areas that TCD was present, but not every single tree was confirmed prior to this study that it has a presence of disease. In California, this is from the orchard where it was heavily infested, so we knew that the disease pressure was much higher. So what we found is we can use our molecular detection. 85% confirmation rate was in California, and we were trying to figure out ways we can confirm Geosmithia morbida much easier versus Geosmithia morbida combined with walnut twig beetle. And then walnut twig beetle was a little bit more challenging in this and the future processes. And we need to work on that part a little bit further. So what we wanted to do is take that approach. So we knew that we had a system that worked pretty well, but we wanted to simplify the process and use basically simple PCR reaction that can be done in disease diagnostic clinic, performed with low input cost and limited equipment or specialized equipment. So we wanted to do several things. So the first one is we developed a method of simply PCR amplification of DNA samples using either Geosmithia morbida or Periopteris juglondis, so the pathogen or the beetle, specific primers, and visualize that simply by running on a gel, so doing regular PCR reaction and running it on a gel. So we did this by basically developing several different DNA sample constructs, and I'd be happy to talk about details of that a little bit later, how we did that and how we mocked everything from pure culture to contaminated wood to diluted DNA in clean wood, contaminated wood. So we did a number of basically mock reactions to mimic what really happens under the field conditions. The second approach is to do the PCR reaction using the TACMan probes and just simply visualize the color change. So the idea is to use regular PCR amplification, the same thing, use TACMan probes that we designed and detect the color change with green, meaning positive, or red, meaning negative. So that worked pretty well. And the last one is we wanted to confirm that this method really works, and we used the gold standard for molecular detection, which is the qPCR amplification using the same TACMan probes, and we know it works, and it's pretty successful, and we published this work in Plant Disease in 2021. So this work was funded by Forest Service NUCFAC. So we wanted basically to utilize this molecular detection and improve our diagnostic. And we know that TACMan probes work. The fluorescence and excitation is pretty successful. It's simplified. All we need is basically high energy blue light and the visualization with the glasses that blocks the blue light. And this can be done basically in any lab without any very specialized equipment. So the nice thing about this, we had a proof of concept that this actually works with not only Geosmithia morbida and G. obscura, which is another closely related Geosmithia species, but we also did a proof of concept that we tested with number of other pathogens, other organisms, animals. So we had around 9 different species that we tested this proof of concept, and it worked. And that was one of the reasons we wanted to extend this to 2 other major diseases that are creating a problem in the eastern United States, which is the second NUCFAC project that was funded using the same approach. One is laurel wilt, and the other one is oak wilt. So this is part of the PhD dissertation of my student, Meher Oni. And the reason we are concerned with laurel wilt, it extends according to the projections beyond the border of Southeast. So it was based on the projection in 2008. It was not even supposed to hit Tennessee at all, but we discovered 2 years ago laurel wilt and sassafras, which is the native range in Tennessee, and disease is spreading across the central and east Tennessee, as well as southern Kentucky. The other issue is the laurel wilt is problematic for us, not necessarily on avocado, which is a big problem in Florida, but problem on sassafras, the native trees, and we think we can utilize the same methodology for this and for the oak wilt that we're currently working on. So having said that, I'd be happy to take any questions after Pramita is done. I assume that's how we're going to go with that.